My name is Kumaran Ramamurthy, and it is my pleasure to introduce, on behalf of the Lambda Lunch Bacteriology Interest Group, Professor Julie Theriot from the University of Washington Department of Biology. Professor Theriot was an undergrad at MIT and received her PhD in biochemistry and biophysics from the University of California, San Francisco. She was then a fellow at the Whitehead Institute for Biomedical Research in Cambridge, Massachusetts, with their own lab for 4 years, and then joined the faculty at Stanford University as an assistant professor, where she rapidly rose through the ranks and became professor in 2012. Last year, she moved to the University of Washington. Professor Theriot has been at the forefront in studying the mechanics of how nanometer-sized macromolecules, such as proteins, self-organize to form micron-scale cellular structures and how self-organizing structures drive cell motility. To address these questions, Professor Theriot has elucidated organizational principles in a variety of model systems, ranging all the way from bacteria to fish skin, employing diverse methods, including biochemical and biophysical approaches to measure and reconstitute complex cellular reactions, mathematical modeling, and genetic and cell biology techniques. For her work, Professor Theriot has received many awards, including MacArthur Fellowship, Packard Foundation Fellowship for Science and Engineering, the American Society for Cell Biology Lifetime Achievement Award, and was named as a HHMI Institute investigator in 2008. For those of you interested in bacterial cell biology, I should mention that Julie will be presenting a second lecture tomorrow in Building 37 in the 6th floor conference room at 10 a.m. titled Unexpected Adventures in the Bacterial Cell Envelope. Please join me in welcoming Professor Theriot.
Thanks very much for the invitation. I'm really happy to be here to speak, meet some great new friends, see some old friends who I haven't seen in many years. And I'm particularly grateful to all of you here in the audience for coming out to hear my Wednesday afternoon lecture on a Tuesday afternoon. What I'm going to talk about today is mechanisms underlying rapid cell motility. And as was mentioned, part of my group also works on bacteria. And here in the illustrations of rapid cell motility, I'm going to show you sort of competitions from the eukaryotic to the prokaryotic world, where the eukaryotes are going to win, because today's all about eukaryotes, but tomorrow I promise the prokaryotes are going to win. So for understanding cell motility, one of the things that I find really fascinating about biology as a discipline is that there's so much variety in terms of the life that we have on this planet. And so if there's some particular cell biological process that you're interested in studying, you can probably go out into the wide world of biology and find some particular organism or some particular cell type that's just really good at the thing that you want to explore. And for me, that's really cell movement. And so we have focused our effort in my group on trying to understand the motility of cells that are particularly good at moving fast, because it seems to put physical limits on what they're able to do that we think is informative in trying to understand the general rules of motility overall. So the first cell type that I'll describe today is the fish epidermal keratocyte, which is shown here on your left. And this is a cell that's derived from the basal layer of the skin of fish or of amphibians, which is specialized for very rapid wound healing. And as you can see, it moves in a very persistent way. It tends to move straight for a very long period of time and move at a constant speed. And so for the biophysical approach that we want to take to understanding interactions among different cellular components during the process of motility, this is a wonderful model system because it essentially operates at steady state. Everything's happening at the same rate so that the cell can move forward without drastically changing shape or changing direction. Now, the second cell type that I will describe is a more recent set of experiments that we've done on human neutrophils and neutrophil-like cell lines. So this movie over here on the right shows an individual cell from a leukemia-derived cell line that can be differentiated into neutrophil-like state, showing the cell crawling around on a microscope slide and phagocytosing Staphylococcus aureus bacteria that are expressing GFP. This experiment was actually done because my student, Lorenzo Lebedigan, when he started work on these HL60 cells, was sharing a bay with a different student, Snow Cho, who was working on cell division in Staphylococcus. And so they decided they would get together and see whose project was stronger. In this particular case, the neutrophils actually won. But for those of you who really want to hear about the bacterial side of the story, that's more what I'm going to be focusing on tomorrow. Anyway, for today's purposes, what I'd like to do is describe a little bit about what we know about sort of the general rules governing this extremely rapid kind of cell motility with rates that are happening on the order of about 0.2 to 0.3 microns per second, or about 100 times faster than a fibroblast would crawl. Starting off with our basic understanding that came largely from analysis of keratocytes, and then branching into the more complex behaviors that we can see with neutrophils. Okay, so in general, part of the reason that I find this problem so fascinating is because of really overall interest in understanding how structure emerges in biological systems. A very small-scale illustration of that would be the protein folding problem, where, as we all know, we can encode a linear string of amino acids in genes that are then able to fold up, sometimes with a little bit of assistance, but fold up into a single stable folded state, or a suite of closely related stable folded states. And understanding how those interactions among all those different amino acids in that polypeptide chain can give rise to a complex three-dimensional structure is a challenging problem that's occupied a lot of very smart biochemists for many years. Now, as soon as your protein chain is folded up, you then have a nice structured protein, for example, the actin molecule that I've shown here on this slide, which is about 5 nanometers across, and it's a typical globular protein that's also able to bind and hydrolyze a single molecule of ATP in the cleft that's shown there at the top. But then what it actually does inside of cells is, of course, vastly more interesting than just achieving that structure, which is catalytically active. Actin is able to self-assemble, as shown in the illustration right below that, by forming a head-to-tail polymer with many individual actin monomers coming together to make a filament, which can be hundreds or thousands of subunits long. And those monomers, their assembly and disassembly is governed by ATP hydrolysis in a complex way, such that cells are not only able to assemble these structures that are hundreds or thousands of times larger than individual proteins, but are also able to rapidly rearrange them, in part in response to internal cues and in part in response to the external environment. And so the large-scale consequence of this is illustrated in the drawing on the right, this beautiful Purkinje cell drawn by Santiago Ramon y Cajal. And in this cell, as you see, the overall size scale is 4 or 5 orders of magnitude larger than the structural proteins that are actually able to make up the filaments. But in order for biological function, the branching and the synapses formed by the cell have to occur in the right geometry. And so somehow, even though that kind of geometry cannot be encoded within the structure of the proteins themselves, because there's an almost infinite variety of shapes that cells can assemble their actin and microtubule cytoskeletons into, nevertheless, the ability of those proteins to self-organize must somehow underlie the ability of the Purkinje cell to develop into this complex morphology. Now, I think that problem is interesting enough, this question of how we're going to be able to discover organizational principles that are able to scan across this extremely large range of structural scales. But I think it becomes even more interesting when we think about this in the context of cell motility, because this really, to me, dramatically illustrates the problem that cells need to be able to not only assemble their internal structures into these large-scale organized arrays, but also be able to rearrange them very quickly in response to changes in the environment. So the movie that I'm going to show here over on the left is another movie of a neutrophil eating a bacterium. This one, though, was made in the 1950s by David Rogers at Vanderbilt University. And here, instead of using an immortalized cell line, this is a primary neutrophil that appears to be in a blood smear, presumably from David Rogers' own blood. And when the movie starts to play, what you'll see is there's a bacterium there over on the left, which it's clear the neutrophil is in the process of actually hunting down. And you can see it pursues the bacterium as the bacterium is bouncing around due to hydrodynamic flows in the system. There's another bacterium that the cell kind of sniffs at, but then it ignores it and stays focused on its original prey. And as it continues to crawl around, you can see it's able to reorganize not just the protrusion at the leading edge, but rather the entire polarity of the entire cell until it's finally able to catch up with the bacterium and engulf it. Now, for those of us who work in the cell motility field, this is considered to be like the greatest movie ever made. It's sort of the Citizen Kane of our field. And I've shown this movie probably 1,000 times, and I hope many of you have also seen it before. Part of the reason I love it so much is because I very happily spend an entire scientific career just trying to understand everything that's going on in that movie. And in particular, what I want to talk about today is this question of how the cell is able to organize movement at its front and movement at its back in order to maintain this single-minded persistence, even when it's getting confusing signals from its environment, and what that must mean for underlying mechanisms. Another aspect of this problem that I find very enchanting is illustrated by this other movie. This is Acanthamoeba castellanii, which is a soil amoeba. that crawls around using the same kind of actin-based motility as we see in human neutrophils, using many of the same underlying proteins and even regulatory mechanisms. And yet, these two cells have been separated from each other by on the order of 1 billion years of evolution. So these processes of rapid directional actin assembly and rapid actin turnover that give rise to rapid cell motility are very widely conserved and very ancient in the eukaryotic lineage, and I think really beg for a large-scale understanding. Okay, so what do we know then about how these molecules can build up to make these large-scale cell processes? Well, in the context of cell motility, we actually know a lot about a couple of specific molecular machines that are involved in this process. So over on the left is a diagram that I'm sure many of you have seen before from a review by Tom Pollard and Gary Borsi, summarizing a lot of information that had come together in the 1990s over regulation of actin filament assembly. So what we see here is a group of actin filaments that are growing in a branched array, impinging on the membrane of a cell. And here I want you to imagine the cell is crawling upwards, just like the movie that I've shown you before. And the basic idea that was summarized in this review article, and which is generally accepted in the field as being, at least a reasonable approximation of what goes on inside of living cells, is that actin filaments grow up against the plasma membrane, and insertion of actin monomers between the tips of those filaments and the membrane as it's fluctuating back and forth are what actually enables the leading edge of the cell to move forward. Now, the birth of those filaments comes about because of the branching nucleation of the ARP2-3 complex that binds to the side of a pre-existing filament and then nucleates a daughter filament off of it, so that all of the filaments are born pointing sort of, but not exactly in the same direction towards the top. And then over time, as ATP is hydrolyzed within the filament, there are additional disassembly factors that come in that preferentially bind to ADP-containing actin filaments and take them apart to regenerate monomers so that the monomers can then diffuse back to the front of the structure and the whole thing can occupy a steady state where it's constantly polymerizing at the front and depolymerizing at the back to push the membrane forward. Over here on the left is a diagram of a different part of the cell. This is actually from the work of Claire Waterman, describing the organizational structure of the molecules within the cell substrate adhesion complex, where as that actin network is turning over, pushing out the front of the cell, it's able to actually translocate the cell body because the actin network is connected to the substratum through these transmembrane connectors, these integrin proteins, that enable forces generated in the cytoskeleton to be transduced in a way that can generate friction against the surface and push the cell forward. And again, thanks to the work of Claire and many other people, we know a lot about the identities of the molecules that are involved in those adhesive structures. We know a lot about their organization, and we know a decent amount about their regulation. But the point I want to make is that both of these detailed molecular diagrams are explaining things that happen over only very small distances in space. So the Pollard and Borsi model for branched actin network growth really only describes what's happening in the first micron or two right behind the leading edge of the motile cell. Whereas really, when the cell is moving, what we see is that there's coordination among all of these molecular micromachines everywhere throughout the cell, such that for this particular keratocyte, the actin that's polymerizing on the left side of the cell is going at exactly the same rate as the actin that's polymerating on the right side of the cell, which is 30 microns away, so that the cell can move straight without changing direction. Furthermore, the disassembly of the actin network at the back of the cell has got to be happening at exactly the same rate as the assembly of the actin network at the front, so that the whole thing can move forward without apparently changing shape. So although we know a lot about coordination of molecules over these scales of a few hundred nanometers or maybe a micron, figuring out how that actually works over these distances of tens of microns or even more in some cell types in order to generate coherent motility, that's still an area where we understand less. Okay, so our general approach to understanding this problem has been to use video microscopy as a primary tool, which I know I have in common with at least a few of the members of this audience. And video microscopy is incredibly appealing because in the modern era, we have not only great ways of visualizing cells, but also great ways of labeling components within cells in order to actually measure directly the dynamics of all of these underlying molecules that are giving rise to our complex behaviors. So this is very powerful. But the flip side of that, which I know also many of you are familiar with, is that a single graduate student can sit down at a microscope and, in a couple of days or a week, can easily accumulate a terabyte of image data on a hard drive. And while in one sense that's very powerful, in another sense that's almost sort of crippling, because once we have all that data, we have an obligation to try to figure out how to actually understand it. And I think this is really beautifully summarized by this quote from Sidney Brenner, whom, as I'm sure you know, we lost just very recently, that we are drowning in a sea of data and starving for knowledge. And so in this context of cell biology, where we can have complex live cell samples generate terabytes or petabytes of image data, the question is, how do we actually extract meaningful measurements from that data in a way that we can get our puny human minds around, that can actually lead to some sort of conceptual understanding of what the rules are that govern these complex behaviors, without losing too much information from all of the effort that we put in technically in order to gather this information. So what I'm going to describe today are two general approaches that apply for the cases I'm going to be telling you about under two slightly different kinds of circumstances. So one general approach is to try to find steady state approximations. And obviously this is going to apply most to the keratocyte system that I showed you before, where the cells are moving with a constant speed and a constant shape. And in that case, the fact that the cells seem to be putting a lot of effort into maintaining a steady state as they're moving along means that we can do things like average measurements over time and even average measurements across different cells in order to try to extract out various fairly subtle dependencies that might not be that obvious if we weren't able to remove noise by averaging. The other general approach that I'll talk about more in the context of neutrophil motility is to find ways to reduce the dimensionality of that data, such that even if we start off with very complex data sets, that we can reduce it down to particular measurements that we believe are biologically meaningful. Again, without losing too much information, but in a way that we can maintain information about how things change over time and about how things change from one individual to another. So I want to start off giving a little bit of background about what we know in terms of just the steady state straight movement of the keratocytes that I showed you, and then spend most of the time talking about more recent work looking at non-steady state movements, both of keratocytes and then of the neutrophils. Okay, so the keratocytes, again, part of the reason they're appealing is because they have a very simple and uniform cytoskeletal organization, which I think is best illustrated in this beautiful electron micrograph from Tanya Svitkina, where in this preparation, she's isolated some of these cells from fish skin and then extracted them and prepared them for platinum replica electron microscopy. So you can see up in the left-hand corner there on the top in part A, that's a whole cell, and then she's zooming in on the actin-rich lamellipodium of the cell in part B, and then zooming in yet further on those two little boxes over on the right side. And what I hope you can see is that the actin filament arrays here are extremely dense. So the separation between actin filaments is on the order of just a couple of widths of actin filaments themselves, so on the order of maybe a couple tens of nanometers. Now, this cell type is really effectively two-dimensional in terms of its lamellipodial structure. If you were to take a cross-section through it and look at it sideways, it would look like a baseball cap, like what you see there at the lower right, where the nucleus and all the membranous organelles are enclosed in that tall cell body that can stand up 6 or 8 microns above the surface. But all of the actin in the lamellipodium is basically in this two-dimensional sheet of felt that is only 200 or 300 nanometers tall and is pretty uniform in its thickness all the way to the back of the cell. You can see this in a slightly different way in this structured illumination micrograph from my student Sunny Lowe, where she's labeled the actin filaments in green, and then the adhesion complexes that I showed you before in the previous slide are these little blue spots that, as you see, are distributed all over the cell in order to connect the dynamics of this actin structure to the substratum. And then I also want to draw your attention to the non-muscle myosin 2 that's shown there in red. And for Jim Sellers, this is almost entirely non-muscle myosin 2A that's present in this cell type. And you can see there are those little red dots of myosin, which we think are of the order of single myosin minifilaments that collect towards the back of the cell and are particularly associated with this transition where we go from the dendritic network that I described with the ARP23 branching into what looks like a much more sort of parallel network of horizontal bundles of actin filaments, which, as I'll show you, are also contractile. So what we really want to understand then is how this large-scale structure emerges and how it can be so reproducible from one cell to another, and also so reproducible in terms of its behavior. So one of the other fun things about the cells that makes them very, very useful with this kind of biophysical analysis came from an experiment done by Ursula Utenauer back in 1984, where what she did was took a microneedle and actually dissected off a piece of the actin-rich lamellipodium from one of these cells. And we see over on the left is a drawing where you can see a whole cell moving at a certain time interval on the top, and then one of these enucleated fragments of the lamellipodium moving at the bottom. And what she saw was that they move with essentially the same persistence and also the same speed as do the whole cells. So we know then that all of the machinery necessary for this persistent rapid motility is actually contained within the lamellipodium itself. You don't need the cell body, you don't need the nucleus, you don't need gene expression, you also don't even need mitochondria or microtubules. So this is a very, very simplified form of actin polymerization-driven motility. On the right here, we're going to see a movie that's a modern reenactment of this experiment that was done by my student Aaron Barnhart, where this is now a fragment of a keratocyte at about the same time dilation as the previous movies I've shown. And you can see it really moves in this beautifully persistent way. There it just crawled over a little piece of schmutz on the cover slip, so it sort of split itself in half. And it's actually going to go through here in complete cytokinesis. So now we have two little daughter cells that are each able to crawl off in their own directions. So this is very powerful in the sense that if we want to understand the mechanics of motility, we want to understand what causes persistence, what causes this directional motion, and what causes coordination among the different parts of the cell. I think this experiment really strongly argues that we really only need to focus on the cytoskeletal elements that are present in that lamellipodial structure, which is a great simplification. So over the years, we and others have developed methods for trying to make quantitative measurements about the dynamics of what's going on in all these cytoskeletal elements. And this movie I put in for Claire. This is one of our early attempts at doing fluorescent speckle microscopy, which was a technique that Claire pioneered, to actually be able to measure dynamics in the actin network as a whole. So in this cell, it's been electroporated with a very small amount of phalloidin, which labels the actin cytoskeleton, but done at a very low level so that you see this sort of texture arising within the network. And for these rapidly moving cells, it's very hard in the lab frame of reference to actually see what's going on with the network. And so typically for most of the movies that I'm going to show you, we will translate them into the cell frame of reference. So as if the cell had a camera that was following it around, and we're seeing the movements from the cell's point of view. So in the cell frame of reference, I hope you can see with the fluorescent phalloidin speckles there that they're sort of raining straight down within the cell frame of reference, which seems to indicate that the actin network, which is assembling at the leading edge, is moving backwards sort of as a coherent unit until we get all the way down to the cell body. And then you can see there's some inward flow on the left and right sides where the back ends of the actin network are being pulled in towards the cell body. But overall, the whole thing is really moving at a pretty constant rate with respect to the cell. And it's actually close to stationary with respect to the substratum, as I'll show you in a moment. You can also see a lot of other details in the cell frame of reference. So for example, over on the left, I just want to point out, if you look at the cell body, you can see that it's sort of rolling as the cell moves forward. That's something that's quite hard to appreciate in the original movie, but probably speaks to the kind of mechanical coupling that's happening between the cell body and the lamellipodium overall. So Goud and Stanuser, working together with people in Claire's lab, worked out some really nice methods for being able to actually track movements of those kinds of speckles throughout the lamellipodium, or in fact the entire cell of a living cell moving like this. And I'm showing you just one of these movies here, where over on the right we've done, again, temporal averaging for the movements of a single cell so we can get some general rules about how this thing behaves. And again, left is the lab frame, right is the cell frame. And as I described before, in the cell frame of reference, the actin network is moving coherently backwards throughout most lamellipodium. But with respect to the lab frame of reference, it's stationary. So what that means is as the actin network is being built, it's being coupled to the substrate through these adhesion complexes pretty much immediately and directly. So further polymerization pushes the leading edge forward, and then those adhesion complexes have to be peeled up at the back as the depolymerization also happens. And you can see in the lab frame of reference, some of that peeling up at the back is coming about because of inward flow of the actin network, which, as I described, is thought to be driven by that non-muscle myosin 2. Okay, so as I said, this is a great model system for doing biophysical analysis. And over the past 10 years or so, my group has essentially dissected this system in its steady state by examining the mechanical contributions of every element of the cell that we think can be contributing to the overall coordination of its movement. So just to summarize again, as I described, we have actin polymerization at the front, depolymerization at the back, formation of adhesion complexes at the front, dissolution of adhesion complexes at the back. There's also this inward contraction of myosin, which has an interesting secondary effect, that because the back of the cell is being squeezed, that sort of shoots fluid through the lamellipodium up towards the front of the cell. And we think that that hydrodynamic fluid flow actually can carry actin monomers and ARP23 complexes and other materials up to the front where they're needed for polymerization. And furthermore, because the whole cell is enclosed by a plasma membrane, which is contiguous over the surface of the whole cell, and which Aaron Barnhart and Theriot have shown is actually under really quite high tension as the cell is moving, that bag, if you will, that's containing this dynamic actin cytoskeleton can act as a mechanical integrator, such that what happens at the cell, the information about changes at the front of the cell, can be immediately propagated to the back of the cell, even over these large, 10, 20, 30 micron distances. Okay, so again, I want to just summarize a couple of key experiments about what we know in terms of how this thing is mechanically driven in order to set up the new work on non-steady state motility. This is a set of experiments that were originated by Cyrus Wilson when he was a graduate student in my lab to really try to look at what governs the overall pattern of actin filament disassembly in these cells. So Cyrus worked with people in Gowden's lab to come up with a really simple way of estimating the distribution, the spatial distribution of both assembly and disassembly of these networks using the data from speckle microscopy. And it's a very simple idea. It's just that if you are imaging something that's moving at steady state and you have some area of the cell where you see a constant influx of the actin network from all directions or from any directions, an influx, a net influx, it must be the case that the actin network is disassembling in that region. And so using that just very simple logic and making a map of the whole cell, as you see over there on the left, the polymerization shown in purple, the net polymerization, is happening mostly at the front of the cell where we would expect it. The depolymerization is happening mostly at the back, but most strikingly, it's happening primarily in these two great big spots that are right on either side of the cell body. And that really intrigued us because we knew from other experiments that those two spots are exactly where we have the maximum accumulation of these non-muscle myosin II filaments. So this movie now, again from the cell frame of reference, shows instead of labeled actin like I showed you before, this is now labeled myosin. And what I hope you can appreciate is that there's sort of an increase in the density of the little dots as you move backwards towards the cell body and the lamellipodium, presumably because of individual myosin mini filaments that are able to get activated and land on the network that then ride the network towards the back. And we were intrigued by this connection between the distribution of myosin and the distribution of depolymerization. Cyrus suspected that that might suggest that myosin was actually catalyzing depolymerization in this system, which he was able to get some support for with pharmacological experiments. But I think the most convincing experiment along those lines is this one, which was done by Mark Tsushita, where he took cells that were crawling along and sort of minding their own business and then extracted their membrane with Triton. So this left the actin cytoskeleton, all its binding proteins left behind, but all of the soluble elements are now removed from the cell, including all of the ATP. And then one of these isolated cytoskeletons that you see on the top there at time 0, he could then add ATP and then watch what happened overall. And we're going to see this in the movie on the right. The addition of ATP to this kind of preparation then causes the back half of the actin network essentially to just melt. And again, using pharmacological inhibition, he could prove that this activity required the enzymatic activity of myosin II. So in other words, what we're suggesting here is that the myosin II at the back of that cell is actually acting to catalyze depolymerization of the network in that particular region. And at the time, this was quite a surprise to us because we had been accustomed to thinking of myosin as really generating mostly contractile activity. But a number of people have shown in vitro, including Jim Seller's group, that myosin actually can act as a severing protein for actin filaments. And we suspect that it's catalyzing the disassembly of the actin network simply through physically breaking them apart in those regions. So this is now our overall model for how we get this large-scale coordination, why it is that the distance from the front of the cell to the back of the cell is able to remain so constant as the cell is moving, is because the myosin incorporation is acting as a depolymerization timer. So we have our little mini filaments that hop into the network at the front. They start riding them to the back. When they first start to assemble on that dendritic part of the actin network, the orientation of the actin filaments is not particularly favorable for myosin to generate very effective force. But as we move backward in the network, as the network ages, those myosin mini filaments, and now there are more and more of them that are arriving, can start cooperating to reorganize the filaments so that they end up in more of this parallel structure that's perpendicular to the direction of movement of the cell. And then when they accumulate and cross some nonlinear threshold, at the very back they get to this very, very high density where they're able to rip the network apart. And that seems to make sense with respect to this maintenance of the steady state. So at about the same time, we started paying attention to the fact that, although part of the reason that these keratocytes are appealing is because all of them look more or less alike in the sense of having a single large lamellipodium and moving in a fairly persistent direction, nevertheless, they didn't look exactly alike. So this, for example, is a rogues gallery of a bunch of keratocytes that are all isolated from the same scale of the same fish. So they're all genetically identical. But as you can see, they have a range of shapes. Now, some of them are big and some of them are small. That's probably not that much of a surprise. But also I want to show you that some of them have this quite wide aspect ratio where they're very long and sort of canoe-shaped. But some of them over more on the left are much rounder. They have almost a circular shape or a D shape. And this, again, is clearly not something that is caused by genetic differences. And we're also able to these variations are persistent, and they're also cell intrinsic. So this suggested that there was some sort of underlying variation in these genetically identical cells that was giving us slightly different shapes and perhaps slightly different behaviors. So in order to quantitate this variation in shape, Zach Pincus and my group came up with a very simple method for doing principal components analysis on shape information from cells, where we're able mathematically essentially to describe what the modes of variation are that explain the most variation within a data set, the second most variation that's orthogonal, the third most variation, et cetera. And this is the pattern that he came up with for the shape mode determination in the keratocytes. So this first shape mode, which describes actually about 80% of the total quantitative variation in cell shape, appears to be just largely correlated with the cell size. The second shape mode appears to be largely correlated with the cell aspect ratio. And those are things that we probably could have guessed just looking at a bunch of cell images like the ones I showed you before. But what this analysis shows us is that those two things really do dominate the variation and also are orthogonal to each other mathematically. Now, looking a little further down, we start to get some more interesting clues, because these are now shape modes that only represent a few % of the total variance, but are very robust within these populations. And the mode number 3 seems to be the position of the wings, whether the wings are sort of drooping or whether they're standing up straight. And then shape mode 4 looks to be left-right asymmetry. So given sort of the persistence, the robustness of these kinds of shape variations, and also the fact that, as I told you, the shape variations are cell intrinsic, we started correlating variations in shape with variations in cell behavior. And the bottom line is that these cells that are very wide also tend to have a smooth leading edge, and they tend to be the ones that move very fast. And so we call those coherent cells. Those are the ones that are really super persistent. But then there's another set of cells, which is actually just sort of the opposite end of this continuous spectrum, that we call decoherent, where they tend to be smaller, they also tend to be rounder, have a rougher leading edge, and also move more slowly. So we worked together with Alex Mogilner to try to put this within a comprehensive biophysical model to try to understand what could be the origins of this variation. And again, just to show you that the outcome of those studies, that first mode of variation, the size of the cell, is something which, although it can vary quite a lot in the population, is something that stays absolutely constant for a single cell as it moves along over at least many tens of minutes. So that's something that's really cell intrinsic. The cell aspect ratio, on the other hand, is something that we can only really rationalize by appealing to the balance between the force that's being generated by these actin filaments pushing against the membrane and the membrane tension that's opposing those actin filaments. And I'm not going to go into the details of this because this was published now about 10 years ago, but the bottom line is that we can rationalize the difference between those coherent cells that are wide and move very fast versus decoherent cells that are narrow and move more slowly, by postulating that the only important difference between those two cell types is the amount of actin that's there. And if you're a cell that's got a lot of actin and it's pushing mostly at the front, and then as you move outward from that center, there's relatively less and less dense actin network at the front, the point where the membrane tension overcomes the force generated by actin filaments and then the cell sort of turns the corner and begins to collapse is quite far away. And so you have a very wide leading edge. Conversely, if you have less actin, then you can't get so far away before it starts to collapse, and therefore you have a narrow leading edge. And Alex was able to put this together into this very simple analytical model that describes the variation in terms of the cell aspect ratio and also the cell actin density in terms of microscopic rate constants describing, as you see there, the T is the tension of the membrane, gamma is the rate of capping of actin filaments, beta is the rate of branching of actin filaments, and F stall is just the maximum amount of force that can be generated by a single filament pushing on a membrane. So within this outrageously simple analytical model, we can rationalize all of these different aspects of cell shape and cell speed just by appealing to that simple ratio as being the main thing that determines variation among cells. So that's kind of an audacious claim that, as we go among this wide population where we see a lot of individual to individual cell variation, it can be attributed just to a single factor, which is essentially how much filamentous actin there is in the cell at a given time. And Greg Allen speculated that if that were true, we should then be able to recapitulate that entire spectrum of shape variation within a single cell if we had some way to simply manipulate how much actin was there over time. And so he did this experiment, which is, again, very simple, just changing the temperature at which these cells are moving. And this is another advantage of these fish cells over mammalian cells, is mammalian cells are like fussy little princesses, and if you do anything bad to them, they just die. Whereas these fish cells, they live on the outside of a fish, and, so they have to be very robust to changes in the environment. So you can change their salt concentration by 2-fold or 3-fold in either direction. You can put them in 10% ethanol. They really don't care. You can also change their temperature over the surprisingly wide range. So in this movie, we're going to start off at about 17 degrees Celsius, as you can see, and the cell is crawling along. And as the temperature now starts to drop, you can see it's going to start going slower, which, again, I think you would expect. But as I hope you can appreciate, as it goes slower, it's also becoming rounder. So its aspect ratio is decreasing until by the time we get down here below 10 degrees, it's really almost a circle that's moving forward. And then now at this point, Greg's going to start raising the temperature again. And as the temperature goes up, you can see the cell is going faster, but it's also getting wider. Okay, and this is exactly what we expect from this simple model, that those two things should be tightly coupled. And we're basically able to recapitulate all of that variation we see in the population just by changing the temperature of these cells as they're moving around. Now, temperature, of course, is also going to control the rates, the turnover rates of lots of other biochemical reactions in the cell. But we don't need to invoke anything more complex than simply changing the rate of actin polymerization in order to explain all of this variation. Okay, so that's the first two modes, the size and the aspect ratio. As I mentioned, there are these two other modes. The third mode, which is the position of the wings, seems to have to do with the balance between myosin contractility and cell substrate adhesion. I don't want to go into that because I do want to spend a little time talking about this fourth mode, which is the left-right asymmetry. And this one seems to be actually a description of how much the cell is able to turn. So the difference in speed between the left side and the right side of the cell that is actually able to make it change direction. So this is now a fun example of a sort of non-steady state behavior that we can begin to try to characterize in these cells where we understand the steady state very well. And the way this project came about actually was Greg just got curious about how straight these cells are actually able to go. So over short periods of time, they look like I just showed you in this initial movie. But you'll notice this is only a few cell diameters, where you can see the cell going straight. And so Greg made an 8-hour-long movie where he looked over a region of the cover slip that was several millimeters in each side just by tiling back and forth over and over again, with the cells at a low enough density to track individual cells over this very long period of time. And these tracks that he found are shown on the right. You can see there are some regions where they go very straight for up to something like 0.5 millimeter, but then there are regions where they tend to go in little tight circles. And for an individual cell, it can switch back and forth between moving pretty straight and moving in these tight circles. And so we started doing some statistical analysis on what these circles are doing, and it didn't really make any sense. I mean, if you wanted to just explore space efficiently, you could do it much more efficiently with a random walk or a Levy flight or really any other kind of trajectory instead of these knots on a string that he saw. So he started trying to mathematically analyze, well, what is it about the nature of these trajectories that can give us a hint into what it actually means as far as the cell behavior goes. So he started simulating some trajectories, starting, of course, with just a random walk, which does not well fit what we saw with the cells. But we wouldn't expect it to, because in a random walk, each step of the random walk is uncorrelated with the previous steps. What we would have expected for most cell types, and what has been described for other motile cell types, is a persistent random walk, where you're not making a random variation in direction. What you're doing is making a random variation in your change in direction, and d theta dt at each step. That also did not represent what we saw. The closest he got, using this very simple approach, was what we call a random change in the persistent directional error, where it's actually a random walk in the error on top of the change in angular velocity. Okay? So in other words, if you start going in a certain direction, you tend to continue going, turning in that same direction, but there's a little bit of error that's superimposed on top of that. And if you just calculate that out and you simulate trajectories, you see like what we see down in the right-hand corner there, which looks very much like this knots on a string pattern. Now, the place that Greg got this idea from is this paper in Current Biology, which is really hilarious. I highly recommend it. So there's this urban legend that if you get lost in the woods, you're going to walk in a circle. Yeah, everybody's heard this? This is like the plot of the Blair Witch Project. Anyway, these people, they decided they would test this experimentally. So they took a bunch of volunteers to this forest in Germany, and I assume they gave them like snacks and water and stuff, and they just dropped them in the woods, and then they told them to try to walk in a straight line. And what you see in the yellow traces there is what happened when people were trying to walk in a straight line on a sunny day, and you see they did pretty well. But the blue traces are what happens when people who are given the same instructions are trying to walk in a straight line on a cloudy day, and they don't have any, sort of external information about what direction they're heading. And what you see is this knots on a string pattern. And then the traces at the lower left, the lower left, are a similar experiment where they blindfolded people and put them in a soccer field. And you can see most of them did the same thing of going straight and then going in little circles. There was one guy over there all the way on the left who had, obviously some sort of intrinsic bias, but most of the time this is what they did. And this was the model they came up with, was that it's this random walk in angular velocity space. So what this put us in mind of in terms of what the cells are doing is essentially that it looks like once a cell starts turning, its most likely thing to do is keep turning the same way, but then there's a little bit of variation imposed on top of that. So Greg specifically pulled out some of these cells that were actively turning in these small circles to look at how they were different from the average cells that we were used to looking at. And this is one of those movies, again, lab frame of reference, cell frame of reference. And what I hope you can see is that the shape is asymmetric. So on the slow-moving side of the cell, that is the side on the inside of the turn, it's actually quite tall from front to back. It's sort of fat, and that's what we call incoherent. So it looks like a slow-moving cell on that one side. But on the outside of the turn, where it's moving faster, that wing is much more elongated, and that looks like a coherent cell. So the cell that's turning actually looks to us like a cell that is, sort of half and half from the population, where half of it is slow-moving and half of it is fast-moving. And that's why it's able to generate these persistent turns. And in fact, if we measure angular velocity and plot it against this fourth mode of cell shape variation, we see this very strong correlation. So as will come as no surprise, this can be at least in part attributed to myosin. As I described, there's myosin that accumulates in the back of the cell. It contributes to the actin network turnover, as well as to this contraction collapse. And what we're able to see is that for cells that are persistently turning, there's much more myosin at the outside of the cell, that is the fast-moving side, than there is at the inside of the cell, which is the slow-moving side. And that's persistent, as you see in the top right. And also, as we inhibit myosin pharmacologically or activate it pharmacologically, we can change the angular velocity of these cells. So it seems like this myosin asymmetry can be a driver of persistent cell turning. We can also do a lot of other sort of quantitative measurements along similar lines. I'm showing you just a few highlights here. If we measure the actin turnover time as a function of position over here on the top right, what we see is that the depolymerization of actin on the outside of the turn is very fast compared to on the inside of the turn. Again, consistent with the idea that whole side of the cell is essentially moving faster. And then I think most strikingly, when we look at the actin speckle analysis, we can see that instead of the network raining straight back the way that it does in the cells that are moving straight, in these turning cells, the actin network actually rains diagonally. So it delivers more myosin to that outside of the cell because it's got a longer distance to move. And that contributes then to faster inward flow of the actin network also on that side. So basically everything about that side of the cell is just going faster than everything on the inside. And furthermore, we can also see how this is reinforced by that connection to the substratum. If we look at the traction forces generated directly by measuring deformations in a flexible substratum, we see there's more traction force generated on the inside of the turn than the outside of the turn. And if we have stripes of a high adhesive zone next to stripes of a low adhesive zone, and cells crawl over that boundary, they'll always turn towards the side of higher adhesion. Okay, so this is giving us now a very coherent framework where there's multiple positive feedbacks involved in this establishment of left-right asymmetry. You can start, if you like, thinking about the network at the front. As the cell starts to turn, it starts raining diagonally instead of raining vertically down. That delivers more myosin to the outside of the cell. Because of that, the outside of the cell has faster actin turnover. Also, it rips up its adhesions faster. And so that whole side of the cell is essentially slipping around relative to the relatively slower moving, more heavily stuck down inside of the cell. And so the cell is able to pivot as it goes through these turns. So this all comes together into what we call this rear wheel steering model. where the motion of the cell as it's changing direction can come about because of multiple reinforcements, multiple positive feedback loops that enable myosin to stably assemble on one side of the cell at a higher density than the other, that then is giving us all of these linked mechanical asymmetries that enable persistent turning. So that seems like a good model in the context of these keratocytes where, they really like to go at steady state. I'd shown you how they move straight at steady state, and here we see they even seem to turn at steady state. But what this doesn't enable us to do is actually try to figure out the order of events, because this is still a steady state, although a turning one. We can't really see whether it's, asymmetry in myosin that gives rise to the asymmetry in actin flow that then gives rise to all these other positive reinforcements, or is it alternatively something happening at the front of the cell that is then being reinforced at the back? So to answer that question, along with other questions about non-steady state motility, and I'm just going to skip this in the interest of time, we've now turned to these neutrophils, these human cells, which obviously do a lot more complex behaviors. They change direction a lot more as they move. And in this movie, we have this neutrophil actually hunting Candida, which is a pathogenic yeast. And you can see as the cell comes in, it's sort of, it's doing a little bit of a sine wave in its motion, and it looks like it's almost going to miss the Candida, but then it turns around and lunges in order to find them. So lots of complexity, definitely not happening at steady state. And now the question becomes, can we apply our same sort of quantitative analysis to try to figure out what's going on in this more complicated system where everything is constantly changing over time? Now, the neutrophils, of course, their behavior is a lot more complicated. They do chemotaxis, which the keratocytes actually basically don't do at all. And their structural organization is a lot more complicated, as you can see in the micrograph over on the left. We can't really describe their shape paying attention only to the actin anymore because the nucleus is such a big part of the overall shape of the cell. Also, the microtubule involvement in neutrophils can't be ignored the way that it can be in keratocytes. And furthermore, if you look at the movie on the right, which is actually in real time, acquired at 50 millisecond intervals, you can see there's also a tremendous amount of very complex hydrodynamic fluid flow that's going on inside of these cells, where you can see these little granules that are really being squirted forward around the nucleus, sort of bouncing up against the barrier of the actin density at the lamellipodium, and basically just everything changing constantly in ways where we can no longer make this steady state assumption. So because we were so used to doing steady state analysis in the context of the keratocytes, Tony Tsai, who was the first student to really tackle this problem, wanted to ask the question of whether there were any measurable features that were held constant or that we could sort of treat as being steady state in a sort of statistical way that could enable us to leverage what we understood about the keratocytes in order to better understand what was happening in these more complex cells. So the first thing he did was just figure out a nice way to extract the cell's shape from movies as it moved along. And he noticed right away that if you wanted to describe how fast a cell like this is moving, if it's also constantly changing shape, that it's actually quite hard to do that just with a single speed because different parts of the cell are moving at different speeds. So what Tony decided to do instead was, over the course of a movie like this, track the change in area. So how much area is protruded at the front, as shown there in green, and how much area is retracted at the back, as shown in red. And so describe speed in terms of sort of microns squared per second rather than microns per second. And what you see on the bottom then is a trace over 15 minutes of looking at this front edge speed in green versus back edge in red. And you can see both of those things fluctuate up and down over time really quite a lot. But they fluctuate basically phase. And so the consequence of that is shown in that blue line, which is actually it's measurement, it's not an axis. That blue line shows you the area, the projected area of the cell. So even though it's doing this complex shape change, it's speeding up and slowing down as we look through this movie, nevertheless, the projected area is remaining absolutely constant. So that's actually a great simplification that now we can take advantage of as we try to do our more quantitative analysis. Furthermore, when we do this principal components analysis of cell shape variation that I described before, we end up with this outrageously simple result, which is actually even simpler in neutrophils than it was in keratocytes. And what we're looking at here is at the top, this is taken from a single movie of a single cell where Tony followed the cell over an hour, took a picture of it every 5 seconds, and then did that shape analysis on all of those frames. And you see there's variation in terms of the front-rear elongation of the cell. There's variation in terms of left-right asymmetry there in the second mode. And then there's also variation in terms of sort of how flat or how pointy the leading edge is. And then he did a second experiment where he took 300 cells, filmed each of them only for 1 minute, taking a picture every 10 seconds, and then did the same analysis on that big population. And what I hope you can appreciate is that those shape modes essentially are exactly the same. So what this means is that if you just take one neutrophil and you watch it long enough, it's going to be able to explore all of the same shape variational space as a whole population of neutrophils will. So again, that is a real simplification that we can really take advantage of, that ergodicity. And that's actually simpler than what we saw in the keratocytes, where things like the aspect ratio were in fact cell intrinsic. Okay, so in order to begin to address questions like how myosin is contributing to this, and whether we are using the same mechanism to turn in the neutrophils, which are turning all the time, as compared to the keratocytes, which are only turning at steady state, Tony also worked out a nice metric for measuring the front-rear asymmetry in terms of myosin localization, which is illustrated there in the top movie. And here now down at the bottom, we see that trace of the protrusion speed in green, which I showed you on the last slide, but then also now the myosin asymmetry shown in the magenta trace. And what I hope you can see here is that, again, they're both going up and down really quite a lot, but now there's a temporal offset where the protrusion speed changes first, and then the myosin localization changes about 12 or 15 seconds later. And that is illustrated for a population, that cross-correlation over there on the right. This is very robust and consistent from one cell to another. So something is happening at the front of the cell, and then we're getting a redistribution of myosin that's happening somewhat later. And this is great because the fact that these cells are changing direction all the time means that we can now at least begin to assign cause and effect. That is, I can't say that just because the change in protrusion happens before the change in myosin localization, that it's the change in protrusion that causes that change. But I think I can say the opposite, that it's not the myosin localization that's driving the change in speed, because it happens later. But we really wanted to understand where this time frame is coming from. Why does it take 12 or 15 seconds until we see the myosin show up at the back of the cell? And in the interest of time, I'm going to skip the detailed analysis here, but for the aficionados, I can tell you that it's not got anything to do with Rho-A signaling or any other small GTPase signaling as far as we can tell. But instead, what we think is going on is that very same mechanism that's operating in keratocytes is also essentially the mechanism by which myosin is getting localized to the rear of the cell in neutrophils. In other words, it's hopping on the actin network at the front of the cell, it's being transported to the back, it's getting more and more concentrated as you get to the back. And here we can see the actin in a turf image over on the left, again in the cell frame of reference, and then these myosin mini-filaments you can see as little spots in the movie over on the right. And so if we think of the myosin as essentially just being a passenger that hops on and off this moving actin sidewalk, if you will, we can describe the distribution of the myosin with this fairly simple pair of partial differential equations that involve the diffusion of unbound myosin, the advection of bound myosin that's riding along the actin cytoskeleton, and then some on rates and off rates. And most of those parameters are things we can actually measure experimentally. And so if we take those parameters and plug them into just this very simple model, what we actually predict is shown on the right, which is that if we drive a variation in cell speed, if we dictate that cell speed changes in a certain way over time, we see the myosin localization changing about 15 seconds later. And that's not one of those things where we can only get that result if we just choose exactly the right parameters. What Tony then went ahead and did was he took the parameters that we had measured from experiments like the photobleaching assay shown above, and then varied those parameters over an eightfold change up or down, and then recalculated what that delay should be. And no matter how far off on any parameters, just the overall geometry of the system dictates that it must be the case that this time delay is going to be on the order of about 10 or 20 seconds. So in other words, even though we're seeing a much more complicated behavior, things that are not operating over steady state, the underlying molecular mechanism we think is still the same between keratocytes and neutrophils, of just the myosin riding on this actin network, this treadmilling actin network, to get to the back of the cell. And again, in the interest of time, I'm not going to be able to go through this, but we know that the myosin affinity, the filament affinity for the actin network, is governed by phosphorylation with a very well-characterized pathway that Jim can tell you all about. And we're able to interfere with this genetically and pharmacologically and actually be able to demonstrate that the way that the localization of the myosin network changes is exactly what you would predict just from that very simple model. Okay, so to end up, I want to show you how this affects turning. And can we actually use this more complicated system to try to either confirm or refute the idea that this myosin asymmetry driven by the actin network flow is what dictates turning in neutrophils as well as keratocytes. Well, I think the most diagnostic thing that we found in keratocytes, which is very counterintuitive if you think about it, is that there's more myosin on the outside of the turning cell. And that was very clear in that case. It's, as I said, counterintuitive, because if you think of myosin as mostly contracting, then you think that you'd be contracting more on the inside where you're sort of turning over rather than on the outside. But here now, if we look at the neutrophils, and here this is a maximum intensity projection of a single neutrophil that's moving down in this sort of sinusoidal fashion. We're looking at the myosin localization at every step in this movie. You can see the bright spot of myosin is always on the outside. And when the cell changes direction, the myosin actually flips over to the other side, but it's always on the outside of the curve. And again, this is strongly correlated with this left-right asymmetry mode, which for the neutrophils is mode number 2. And again, we can rationalize all of this in terms of it being the myosin localization that's reacting to changes in the direction of the actin network flow that's enabling this large-scale coupling between the front of the cell and the back of the cell, not only in terms of steady state forward changes in keratocytes or constant turns in keratocytes, but also in terms of these massive fluctuations in speed in terms of neutrophils. We have the back responding to the front in this way. And then also the directional changes in neutrophils also seem to all use the same underlying mechanism. And then I'll skip this. But we're doing other stuff. So what I want to end by emphasizing is this idea that there are really dramatic, overwhelming similarities between the underlying mechanisms driving large-scale coordination in keratocytes and neutrophils, even though their behavior is very different. And in both cases, it's the cooperation between actin and myosin that's giving rise to these large-scale behaviors, where cell shape and cell movement in both cases are basically driven by the mechanical interaction among all of these elements that are following the same rules in these two different cell types, where there's only really quantitative changes in the nature of their dynamics that gives rise to these large-scale behavioral differences. So in both cases, myosin is accumulating at the cell rear due to actin network transport, and in both cases, it's facilitating the reorientation of the cell rear in response to that change so that the cell can essentially pick up its tail when it decides to move in a new direction and thereby maintain an overall coherence of shape even as it's changing direction. And importantly, the keratocytes do all of this at steady state, whereas in the neutrophils, for interesting dynamical reasons, seem to be in a more unstable part of this phase diagram where they accumulate things faster and then destroy things faster, enabling them to change direction frequently, whereas for the keratocytes, it's something that happens only fairly rarely. So to come back to Sidney Brenner, I think it's true that we're drowning in a sea of data, but the good news is that these highly complex cell biological behaviors may actually exist in an intrinsically simple and low-dimensional space. And taking these quantitative metrics seriously, we're able to derive ways to analyze these complex behaviors that nevertheless seem to represent a really simple fundamental perspective about the way that this coordination has to happen at a very large scale that may be general for at least these two rapidly moving cell types, the keratocytes and the neutrophils, will probably have to be modified as we start looking into other cell types that move overall more slowly, but nevertheless seem to be things that are fairly conserved in the context of rapid cell motility. And another thing I just want to emphasize is that for all of these projects, we've really taken advantage of the fact that not only can we go in and perturb our cell biological processes using genetics and pharmacology and sort of the standard cell biological approaches, but we can also really take advantage of the fact that there is intrinsic cell-to-cell variation in any cell population that, if you analyze it carefully, can give rise to a fundamental understanding of what the molecular mechanisms must be that govern that process that you're observing. So I'll stop there and thank the tremendously talented group of students and postdocs that I've had the privilege to work on this project with over the last 10 years. Coherent and Zach Pincus started off with the shape analysis in keratocytes, working closely with Alex Mogilner to develop that steady state model. And that work was carried on then by Aaron Barnhart and Greg Allen, and Greg did all the work on turning that I described. Tony Tsai initiated the neutrophil project in the lab, and then that project was further carried on by Caleb Chan. And finally, I'd like to thank all of our wonderful collaborators and also funding sources, and thanks to all of you for listening. Thanks.
Hi, Julia. I realize this is probably in that sort of massive paper that just came out in April, which I haven't had a chance to read. But do you get a sense of which of the two primary kinases that phosphorylate myosin may be driving the polymerization in the neutrophils?
We didn't really try to disentangle them. So we did use a Rho kinase inhibitor, the Y compound, that did have an effect of the myosin localization in terms that, you know, the treatment gave you more delocalized myosin as you would predict. But we didn't really disentangle the effect of Rho kinase from the effect of myosin light chain kinase. I think in keratocytes, at least in the keratocytes that we isolate from embryonic zebrafish, we have data suggesting what's been also shown in other cell types, that the spatial localization of the MLCK activity and the Rho kinase activity seem to be segregated, such that the Rho kinase is primarily acting sort of in the region of the cell body, and the myosin light chain kinase primarily in the melopodium. I have no reason to think that that's not also happening in the neutrophils, but we haven't actually directly tried to compare effects of those two different.
Have you looked at any
kinases.
calcium levels, you know, in real time to see whether there's any correlation there?
Yeah, so we haven't done that in the neutrophils yet, and in part that's because in keratocytes, actually before Greg started this turning project, his project was supposed to be about calcium spiking in keratocytes, and it was incredibly frustrating because he could get beautiful movies of calcium spiking in keratocytes, and sometimes it was periodic and it would change and all that kind of thing. We could not find any correlation between the spiking of the calcium and any cell behavior that we could measure, or, you know, even for cells that were doing, you know, sort of periodic changes in their behaviors, which we could measure under some circumstances, we still didn't see any correlation between the period of the calcium spiking and the period of the cell behavior oscillations. So, you know, clearly there is something going on with calcium in the keratocytes, but we were not smart enough to figure out what it was. So I've been a little just scared to try to look at it in neutrophils, so we haven't done any of that yet. Ed.
So I love your idea that the variation space is small, even though the parameter space is huge. And so we also been trying to think about how you get it to be that way. Do you think it's something about something complicated, like a lot of built-in feedbacks to keep things sort of in sync? Is it the intrinsic structure of the signaling pathways, the convergences that force that? How do you think about how you keep that variation space small?
Yeah, I think that's a really interesting question. I think there's a lot of different aspects of cell biology where this kind of thing shows up, where, you know, you can have a lot of variation, like in terms of signaling inputs, that then, you know, go through some relatively small number of internal pathways, you know, to change cell states, for example, in a different domain. And you know, I think you can either think about it sort of functionally, or you can think about it in an evolutionary sense. In a functional sense, I think if, you know, you have a complex behavior that you want to change, it's important to distinguish between the driver, you know, between the engine and sort of the steering wheel, if you will. And once you have an engine that's operating in some particular way, so in this case, it's really just the polymerization, depolymerization cycle of actin, you know, that's happening in directed sense. Once all of that is running, then you can do anything you want by just tweaking it a little bit here and there, by tweaking it spatially, by tweaking it in terms of its environment, by tweaking it over time. And so it's very parsimonious, if you will, you know, once you have an engine that you've built to figure out, you know, modulations that all come in and operate on that same engine, where maybe the number of adjustments that you can make, the number of knobs you can turn is really not that large, because the underlying engine is just fairly simple. And, you know, I think in this case, it's hard for me to really argue convincingly that that's got to be the case for actin-based motility, because, you know, all of our analysis has been so actin-centric that it's very easy, you know, to sort of drive everything in through these actin-centric lines. But then I would also say, you know, that even when we start looking at complicated behaviors, and, you know, I didn't get into phagocytosis here, but phagocytosis is sort of an interesting extension of neutrophil motility. We don't see anything that we can't at least sort of rationalize in the context of being quantitative modulations of a fundamental intrinsic actin polymerization turnover, you know, kind of machine. And just the fact that the whole thing is so widely conserved in evolutionary terms, as I mentioned, going all the way back to, you know, acanthamoeba and dictyostelium, is also, I think, consistent with that, in that, you know, once you got something good, you know, don't mess with the thing that works, and, you know, just sort of tweak it around the edges rather than changing the underlying mechanism. But that's a philosophy that I think I choose in order to keep things relatively simple.
All right, so please join us around the corner for cookies afterwards, where we can ask Julie more questions, and thank you again.
Thank you.
That's great.